Institution for Science, which is based at Stanford. In 2015, I started a company called iCultivate. The purpose of iCultivate was to cultivate new research-based relationships between academia and industry. I felt that there is a lack of fundamental basic research in industry, which is hard to do. It's really hard to have funding and also resources to do basic and fundamental research, which is done really well in universities. So why not then collaborate between academic and industry sectors and develop new programs where we can do this fundamental research and to understand mechanisms of action, how biostimulants act or any fertilizer acts. So the company provides independent research for this purpose. And one of the products that I will be talking about is developed by Vital Earth Resources. So the research is all independent. So let's jump into it. Why do we do it anyway? So if you are a grower, you're challenged by so many options. Even if we walk around the exhibition hall today, there's so many products. And we do really well in marketing. Marketing is, we excel in that. So every marketing person will come and say, well, you know, my product is going to give you the best results. If you're a grower, you're told every product is better than every other product. So how do we start to figure out what is going on? And how can a grower select and decide how to spend their money? What is the most efficient way to grow their products, to grow their crops and get financial returns? So one of the reasons iCultivate exists is to provide a collaborative platform. So the research is done in a broad collaboration with many researchers. We are based at Stanford, but we have collaborators at Berkeley, at UC Santa Cruz, Arizona State University, and I'll talk about a little bit about the Contra Costa, which is a community college. So all the research is done through collaborations, and this is why it's independent, and the goal is to publish through peer-reviewed process so that we can start to understand the mechanisms of different fertilizers. And the work is done, which is really fun, by community college students. So I started this program with Dr. Polikowski at the community college, where she had a biotechnology course. And these are community college students. The program is called BEST. It stands for Biotechnology Education and Specialized Training. So community college students do the work. They are trained to do science towards publications. They become co-authors on publications, and I think this is really great for workforce development, also career opportunities and networking that they can develop, because some of the work is done at location at universities through collaborations. So far this program has been running for 2 years, and iCultivate has been around for since 2015. So we are hoping to expand and explore more opportunities to do research. What I'm going to talk about is about steroids, which are one of the biostimulants. And the reason I thought to talk about these in the context of human health is for us to think about what biostimulants are, which are very different from fertilizers. So let's look at steroids. Steroids in the early 1970s were known as brasins, and they were discovered in Brassica napus and in the pollen. And since then, we have now more than 70 steroids known. Earlier, Jing and Julia showed all the different hormones and steroids. They also mentioned, I'm showing a structure of brasinolide and homobrasinolide here, but Julia and Jing also showed how all the other phytohormones, which are 9 of them, are present in plants. And they told you that there were like over 130 of one category, or there's so many different phytohormones. One interesting thing about steroids is that they interact with all the other hormones. You can see all the hormones in the red, auxins, stimulactones, and gibberellins. BRs, steroids, interact through pathway interactions with almost all of those hormones. And they impact plant growth from early stage throughout the plant's life to a later stage as well, including stress responses, biotic and abiotic stress. And we know quite well how brassionosteroids act. So brassionosteroids, on the left you can see without brassionosteroid, a cell, and on the right you can see, sorry if I get closer it gets really loud. On the left is without brassionosteroids, on the right is with brassionosteroids. And in a cell you can see the receptor, the BRI receptor, is separated from back. But when brassionosteroids are present, the two receptors combine, and the signal is transferred through phosphorylation, which are the yellow P's, and through a cascade of different proteins, which ultimately leads to what we know as BZR1, is pronounced as BZAR1. So BZAR1 is a transcription factor, and in the absence of brassionosteroids, it's not regulating gene expression, but in the presence of brassionosteroids, you can see BZAR1 and BES1 bind to target promoters, and they regulate gene expression. So brassionosteroids, even though they are steroid-based hormones, they act through gene regulation. So they have a whole signal transduction cascade, and we understand this really fully well. Now, brassionosteroids, like I mentioned, they impact many aspects of plant growth and development, including pollen development, seed germination, every aspect of plant, including flowering, fruiting. So they have an impact throughout the plant's life cycle, and they also are involved in stress responses, both biotic and abiotic. So biotic would be fungi, nematodes, other microbes that can cause stress, disease. On the right-hand side are abiotic, which can be drought, salt, and so on. So they play a very central role in plants' response and growth. And amazingly, they work at really small concentration. So this is 0.01 to 100 nanograms per gram, which is like 10 picograms. That's like 12 zeros after the point, right? So it's a very, very small amount is how they act. This is important. And the other important part is I mentioned that they are steroid-based. So if you look at animal and human steroids, you can see that both brassionosteroids, which are in the plants, and the animal and human steroids have a very common biosynthetic pathway, which separates at squalene 2,3 epoxide. Plants go in a different direction through a different enzymatic reaction. So they are slightly different from animal and human steroids. And structurally, you can also see the lower ones are animal, as we know, progesterone, testosterone, MGA, these are animal-human steroids. The ones on the top are plant brassionosteroids, brassionolide, homobrassionolide, and norcastosterone. So these are different structure. Why is that important? It's important because of the way they act. So if you look at animal steroids, they act through binding the estrogen receptor. And that has been linked, if there is too much of steroid, we know that it can cause issues or problems and stress. Whereas brassionosteroids have a signaling mechanism, like I just described, through receptor kinase phosphorylation activity. So they work at very low concentrations and they have a very distinct pathway. And in general, rapeseed is one of the plants that has high levels of brassionosteroids. And in many countries, traditionally, it's eaten unprocessed. It's also used in tea, and the pollen have been found to have anti-cancer properties, especially for prostate cancer. And the references, there's lots of literature on this. In mice, studies have been done orally, where up to 20 or 60 milligrams per kilogram per day for 24 days, brassionosteroids have been given. And it's been shown to increase appetite, body weight, and even high doses up to 1,000 milligrams per kilogram for 15 days has been found to be safe. But we don't know how these phytohormones or these plant steroids act in animals or humans. We are still studying how they actually function. But what we do know is the amazing amount of research that's coming out. There are positive effects to reduce breast cancer, prostate cancer, colon cancer, lung cancer. And all these are reviewed in the two references that I gave below, but there are multitudes of papers that show that brassine steroids have these positive effects. And they're continuously being tested more, and so the advantages are low toxicity and they're natural plant substances. As was mentioned earlier, there are also synthetic forms, but natural substances like brassine steroids can be used for human health. And in humans, they are shown to be chemopreventive and anti-angiogenic, as well as antiviral and anti-inflammatory. This particular brassine steroid, epibrasinolide, has been shown to be good for to prevent Parkinson's disease as well. It's neuroprotective. So with all of these positive effects on human health, we can start to think in terms of the future of not just brassine steroids, but biostimulants. The reason biostimulants are interesting is because they're not nutrients. They work at low concentrations. So I think we can think about human health. But of course, one of the things that we use biostimulants for is for agriculture. Looking at tomatoes grown in the greenhouse, we can see this particular product is Vitazyme. It's an independent research, and we can see that it increased tomato count and also over 32% increase in tomato number and also weight. So we've looked at it with several other crops as well, and seems to have positive yield effects on multiple crops in the greenhouse. So these benefits of, especially these products can be foliar sprayed. So like someone was discussing, some of it may also end up in the soil. It can be also sprayed in the soil. Because these are phytohormones and they work at very low concentrations, it is easy to apply them. The key question remains, are they active? Are they present in, so this particular product that we are looking at, we did LCMS at Stanford, and we found, if you look on the top, those are the references, brasinolide and homobrasinolide. Those are standards. We can see the peaks of brasinolide and homobrasinolide, and without the standard or the product, we don't see those peaks. But when we look in the product, we find specific peaks for brasinolide and homobrasinolide. So we can detect brassine steroids in the product, and we can see positive effects. Now, I'm mentioning that they have health benefits, but I do not recommend that you go out and drink a biostimulant. What I think we should think about is when we spray our plants, our crops, with different fertilizer and different nutrient-based or non-nutrient-based products, it is important to think about the impact on the environment and also consumption of different, and there's lots of discussion on that topic as well. So it's good to think about that aspect, as well as perhaps the future of biostimulants moving into human health aspects, because there is a lot of research going on here. Just for this talk, I reviewed over 35 publications between those years. None of them showed any major concerns, and they are continuously, especially brassine steroids, are continuously being studied. Plus, most of the studies currently are in vitro. So we are hoping that more clinical trials and more in vivo studies will bring biostimulants into a new light for us to look towards human health aspects. So just to end with this thought that biostimulants are not nutrients, they promote growth, they reduce stress, and possibly they have human health benefits as well. Okay, so as we do the research and we do publications, we also give opportunities for published work to be discussed on a podcast. We have Data Science and Innovating Ag. You can go and listen to more of this particular product and also other products where we discuss the publication and in general how the product came to be. And I think especially with interest in biostimulants, it's a good way to educate growers and as well as other people to learn more about different types of products. And we are always open to invite you as a guest. If you have something to share, you can reach out as well. Okay, so ultimately our goal is to have a healthy planet and healthy life. I'm happy to take any questions if you have.
Thank you so much. We do have questions, and we do have time for questions, and Patrick was the first to put his hands up. The microphone's oh, sorry. But we have somebody else.
Hello.
Patrick, sorry.
I can see you.
It was a good presentation. One question I have is in general biostimulants for a regulatory purpose, right? With steroids, do you think that they will be regulated differently?
For different applications, yes. So for example, for human health and anti-cancer, that has to go through clinical trials, so there are completely different regulations. But I encourage you to come also on Friday. There will be a talk from CDFA, California Department of Food and Agriculture. And I think there are some exciting news that they will share about the future of different categories and regulatory processes. Biostimulants are becoming very popular, as we can see. Steroids as phytohormones used at low amounts are considered as biostimulants. Slowly there are going to be other aspects opening up into biostimulant categories. So one regulatory process is for agriculture, but then it's completely different for human application.
Thank you.
Thank you very much for a nice talk. Patrick Dujardin from University of Liège in Belgium. Maybe I missed the point, but have you evidence that by applying steroids or by stimulants containing steroids, there is actually an increased level of steroids within plant tissues? Because this is of course a prerequisite if you want to propose this as an enhancer of human health. So what is the picture?
Yes, so thanks for the question, because before I answer that, I also want to mention, related to your question, there are steroids that can be synthetic, but the ones that we've been looking at, the particular product we have is a natural steroid, so it's not synthetic. Secondly, when we apply steroids into to plant growth, it's not that the steroid levels are increased in the plant itself. It's being applied extrinsically, and so thought will be that it will go and activate or regulate other downstream genes and help the plant grow better, right? Then on the same side, as a phytohormone, what we can see is that it has additional benefits in pharmaceutical applications. So in terms of biosafety, how safe different products are. If somebody has a choice between applying a synthetic form of a stimulant versus a natural form, and so I think the idea is twofold. One is of safety, biosafety on the plant, which is going to have its impact, and secondly, a second application which is in human research. And as those two meet, I think it provides a broader acceptance and understanding and comfort level to be able to go towards biostimulants to apply in agriculture. I hope that answered your question.
Do we have any more questions for our speaker? If not, can I ask a quick one?
Sure.
The brassicas are also highly producers of glucosinolates. Is it easy, and your extracts that you're working on, did I assume correctly that they're extracted from brassicas, correct? Or not necessarily?
They can be, but not the ones that we work with.
Okay, because it would be a little difficult to disentangle the effects from the glucosinolates and the brassica steroids, probably,
Yes,
right?
yes. There are other natural, so microbes also have, also have steroids.
Mm-hmm.
So there are different ways to access steroids for this application from natural source.
Well, thank you so much to all the speakers of this morning. I think they were excellent talks, and now we move to lunch.